Alright, so today we're going to talk about the lac operon. So recall that an operon is a group of genes that's regulated by a single promoter and single terminator. The resulting mRNA would be a single mRNA, which would be further translated into different proteins. For the lac operon, there's a lac promoter and a lac terminator. In between, there are three different genes, which encodes for proteins that are able to break down the lactose to produce energy. And there's also promoter sites where the RNA polymerase would bind, and operator site where this lac repressor would bind. And this lac repressor, it's encoded by this lac-I gene, which is outside of the lac operon, and it's regulated by its own promoter, I promoter. Alright, so there are two types of regulation for the lac operon. The negative regulation is mediated by the lac repressor, and the positive regulation is achieved by the cap activator protein. So for the negative regulation, it's the concentration of lactose plays a key role here. So as the lac-I gene is it's associated with its own promoter, it's consistently being transcribed and further translated into lac repressor. And this lac repressor is able to bind to the operator region and block the RNA polymerase here from moving downwards to transcribe the lac operon, so that the lac operon expression level is low under this case. However, when the lactose is present, it would be broken down by the beta-galactosidase already present in the cell into allolactose, and this molecule is able to bind to the lac repressor and inhibit it binding towards the lac operon, lac operator, so that the RNA polymerase is not blocked here, and it's able to move downwards, and the lac operon expression can be achieved. However, the expression level of the expression level here by the RNA polymerase is not high enough. It also has to be facilitated by this protein called cap protein. So, however, this cap protein is also it's also only active when it's bound to this CAMP molecule, and this CAMP molecule is synthesized by this enzyme called adenylate cyclase. And through positive regulation, the glucose concentration can further affect the cap protein activation. So when glucose level is high, the glucose is able to inhibit the function of adenylate cyclase, and in turn there would be low concentration of CAMP, and the cap protein is not activated. And when glucose level is low, the adenylate cyclase is not inhibited, and the concentration of CAMP molecule is high, and thus the cap protein is activated. And the cap protein facilitate the RNA polymerase binds to the facilitate its binding towards the promoter region of lac operon, and in turn the expression of lac operon will be increased. So now let's look at the different circumstances when the concentration of the lactose and glucose changes, and how the expression of lac operon might be affected. So when the concentration level is high for the lactose but not for the glucose, the lac repressor would be inhibited by the allolactose, and also since the glucose is not present, the CAMP is able to synthesize, and the cap protein is activated. So this is when the the highest rate of transcription would occur, and so that the bacteria is able to break down the lactose efficiently. And however, when there are no lactose or no glucose present in the surrounding environment, even though this the complex is formed, the lac repressor is not inhibited, so it binds to the operator region, so that the RNA polymerase is not able to express the lac Operon. This actually saves energy for this for the bacteria, because they don't need these molecules for breaking down lactose, as there's no lactose present. And under the circumstances when there's high lactose and glucose concentration, the glucose is easier to be broken down than lactose, so the bacteria would prefer to breaking down glucose. So in this case, the transcription rate is low, because the lac repressor, even though it's been inactivated, the cap protein is also inactivated. So there are low level of expression, and they are able to break down the lactose. But on the other hand, the glucose are more preferably being broken down. And finally, when the glucose concentration is high and the lactose concentration is low, this is when the cell would make their best effort to break down glucose. In this case, the cap protein is inactivated and the repressor is activated, so that the expression of lac Operon is reduced to its minimum level, and the transcription would be very, very low due to lack of the binding, along with the binding of the repressor.